Welcome to the deep dive. We often talk about aging, and one thing that keeps coming up is the epigenome, those little chemical tags on our DNA.
Yeah, the ones that control which genes are on or off.
Exactly. And a big one there is DNA methylation.
Yeah.
We know it changes as we get older. It's actually so predictable, scientists have made these epigenetic clocks based on it.
Right, these biomarkers that try to estimate your biological age from methylation patterns.
And if you caught our longevity paper show a couple of weeks ago, we touched on an abstract.
Oh, yeah, I remember that one. Sounded really interesting.
Well, we decided we had to dig into the full paper. Feels pretty significant, especially for researchers, in the longevity biotech space.
Definitely. It tackles a really core question.
Okay, so the paper is Epigenetic Editing at Individual Age-Associated CpGs Affects the Genome-Wide Epigenetic Aging Landscape. Long title.
Yeah, they often are.
It's by Sven Liesenfelder, Mohammed H. Elsafi Mabrouk, Jessica Iliescu, and, well, a whole team. Published in Nature Aging, March 24th, 2025. So the big question they're asking is essentially, if you go in and deliberately tweak one of those methylation sites known to change with age, does that cause ripples? Does it affect the rest of the epigenetic landscape?
Which is crucial, right? Are these changes just like markers on the dashboard, or are they actually part of the engine driving aging? If tweaking them does cause wider effects, maybe we can influence the process.
Exactly. So how'd they even start trying to answer that? They began with HEK293T cells.
Ah, yes, the lab workhorse. Human embryonic kidney cells, basically engineered to be easy to grow and handle.
Right. So good for a first look because they're standardized, homogeneous, lets you see if your tools work and if you get those broader effects you're looking for.
But we should probably mention they're not a perfect model for, actual human aging. Their own epigenome is a bit different.
Good point. They're a starting point, a test system.
A necessary one, though, to test the principle.
So the tools they used are fascinating. CRISPR-based stuff, but not for cutting DNA, for editing the epigenome, adding methylation.
Right, epigenetic editing.
They had two main ones. First, DCAS9 DNMT3A. So the DCAS9 is like a GPS, takes you to the right spot on the DNA.
But it's dead Cas9, so it doesn't cut.
Right. And it's hooked up to DNMT3A, which is an enzyme that actually adds the methyl tag. Pretty neat.
And the other one?
CRISPRoff, a bit more complex. Also uses DCAS9 for targeting, but it brings in other factors like a KAB domain to establish methylation that's supposed to be more stable, more long-lasting.
Okay, so two different ways to try and add these methyl marks. Where did they aim them first?
They targeted a region in the PDE4C gene. It's got lots of these CpG sites.
Which are the hotspots for methylation, C followed by G?
Yep, and PDE4C is known to get more methylated as people age in lots of tissues. Its level in blood is a pretty good marker for age.
Makes sense as an initial target, then.
So they pointed their tools at this PDE4C spot. What happened? Well, they measured methylation levels, pyrosequencing, alumina bead chips.
Standard techniques?
And they saw a big jump, up to about 40% increase in methylation right where they aimed. So the tools worked.
Okay, proof of principle. Both tools could add the marks effectively at the target.
But here's where it gets interesting. They looked closer using something called bisulfite amplicon sequencing. Lets you see individual DNA strands.
Gets you higher resolution.
And they found that even within the target region, looking at the 26 neighboring CpGs, the methylation wasn't uniform. Some neighbors got more methylated than others.
Huh. So it wasn't just like flipping a switch for the whole area. Suggests some local complexity, maybe sequence context or chromatin structure playing a role?
Exactly what they were thinking. It's not just a blunt instrument. And another thing, this added methylation, it was really stable.
How stable?
They checked over 100 days later. The cells had divided many times. The editing tools themselves were long gone. But the extra methylation at PDE4C was still there.
Wow, over 100 days. That's impressive persistence in the cell culture, especially with the CRISPRoff system, maybe.
Yeah, it suggests you might be establishing some kind of lasting epigenetic memory, which is, pretty important if you're thinking therapeutically down the line.
Definitely. Okay, so the local editing worked. It was stable and a bit complex. What about those wider effects you mentioned, the ripples?
Right. So they zoomed out, looked across the whole genome, and they found what they called bystander effects.
Bystander effects. Sounds intriguing. What does that mean?
It means they saw significant methylation changes, increases and decreases at thousands of other CpG sites, far away from the PDE4C gene they originally targeted.
Thousands. Okay. That is a ripple effect. So tweaking one spot really did have widespread consequences.
Yeah, and the cool thing was they saw similar patterns of these bystander effects with both editing tools, the DCAS9, DNMT3A, and the CRISPRoff.
Oh, that's important. Makes it less likely to be just some weird artifact of one specific tool. Suggests it's a real biological response.
Exactly. It's reproducible.
Yeah.
And they dug into where these bystander changes were happening.
Any patterns emerge?
Yep. The sites that gained methylation as bystanders, they were often in CpG islands or right next to them, the CpG shores.
Okay, so specific genomic regions seem more susceptible.
And even the DNA sequence around the bystander sites seemed to matter. Like a preference for G or C right next door, and T or A 2 bases away.
Suggesting maybe the editing machinery itself, or perhaps repair processes, have certain sequence preferences when these off-target effects happen. Gives clues to the mechanism.
Could be. Now, here's maybe the most fascinating part of the bystander story. They linked it back to aging.
How so?
They checked if these bystander sites overlapped with other CpGs already known to change methylation with age in people, specifically from a big study on blood samples.
Okay, comparing their results to known aging markers.
And they found a significant enrichment. The bystander sites that gained methylation were much more likely to be sites that also normally gain methylation with age in humans.
Wow, okay, so editing one age-linked site preferentially affects other age-linked sites. That really strengthens the idea of an interconnected network, doesn't it?
It really does. It's not just random noise across the genome. It seems targeted, in a way, towards the aging epigenome itself.
So what could be driving this connection across the genome? Did they look into, like, chromatin structure, how the DNA is packaged?
They did. They used ATAC-seq. That's
Yeah.
a technique to see which parts of the genome are open or closed, accessible or not.
Right, mapping chromatin accessibility.
And they found the bystander methylation changes tended to happen more in those open, accessible regions, which kind of makes sense. Easier for enzymes to get in there.
Okay, and what about sites that normally lose methylation with age?
Those tended to be in more closed chromatin regions. So accessibility plays a role, but they also said it didn't fully explain everything. It wasn't the whole picture.
Fair enough. Biology is rarely that simple. What else could connect distant sites? The 3D structure.
Exactly. They looked at that using 4C sequencing.
Ah, chromosome conformation capture. Lets you see which bits of DNA are physically close together in the nucleus, even if they're far apart in the linear sequence.
Precisely. They mapped the 3D interactions for their original PDE4C target site and also another age-associated gene, FHL2.
And did the interacting regions show bystander effects?
Yes. They found that bystander methylation changes were significantly more likely to occur in regions that were physically interacting with the targeted PDE4C site in 3D space.
Okay, that's compelling. Suggests the physical looping and folding of DNA brings distant sites together, making them susceptible to these shared changes, like epigenetic neighborhoods.
Yeah, that's a good way to put it. And they also found that age hypermethylated CpGs, those sites that gain methylation with age, were generally enriched within these 3D interaction networks, the interactomes of the sites they tested, PDE4C, FHL2, MIS1, AS3.
So the parts of the genome involved in aging seem to be physically clustered or interacting, leading to these coordinated changes.
It strongly suggests that, yeah, a 3D network influencing epigenetic aging.
Okay, this is building a really interesting picture. So after seeing these effects from targeting one site, did they try hitting multiple sites at once?
They did. That was the next logical step. If it's a network, maybe targeting several nodes would have a bigger impact.
Makes sense. What did they target?
They picked 5 regions known to gain methylation with age, PDE4C, FHL2, ELOVL2, KLF14, TED1, and 5 known to lose methylation with age, CL1A1, AKP8L, CSNK1D, MIS1 AS3, IGSF11.
Okay, hitting both sides of the age-related changes.
And they tried this multiplexed editing, not just in the HEK293 T cells, but also in primary human T cells and mesenchymal stromal cells, or MSCs.
Moving into more physiologically relevant cell types, Why those specifically?
Well, the T cells are important because, as we said, a lot of the initial age methylation links were found in blood, which has lots of T cells.
Right.
And MSCs are involved in tissue repair, regeneration, and they definitely show age-related changes, including senescence. So testing in different cell types helps see how general these effects are.
Good strategy. So what happened with the multiplexed approach? Let's start with HEK cells again.
In HEKs targeting the sites that gain methylation, they did get some methylation changes at the targets themselves, but fairly modest this time.
Less efficient when targeting many sites, maybe.
Perhaps. And surprisingly, they didn't see significant bystander effects or enrichment at other age-related sites in these specific multiplex experiments. They saw some gene expression changes, but no clear correlation with the methylation.
Huh. That's different from the single target results in HEKs. What about targeting the sites that lose methylation in HEKs?
That was also a bit odd. They saw some initial gains in methylation at those targets.
Gains. Even though they were targeting hypomethylated sites, maybe the CRISPRoff system initially adds methyl groups regardless.
Could be part of the mechanism. But anyway, those gains were temporary, gone by day 15. However, early on, at day 3, they did see bystander hypermethylation enriched at other age hypermethylated CpGs.
So even a transient change at the target could trigger some wider, albeit temporary, network effects. Interesting.
Now, on to the primary cells, the T cells. What happened there?
This is where it gets potentially more relevant for actual aging. Yeah.
Okay, targeting the age hypermethylated sites, FHL2, KLF14 showed effects here, resulted in stable methylation changes at those targets after 21 days.
Good. And the bystanders.
Yes, they saw bystander effects, both increased and decreased methylation, and crucially, these changes were enriched at other CpGs known to change with age in blood, particularly strong enrichment at sites that normally gain methylation with age.
So the network effect seems robust in primary T cells when targeting hypermethylated sites. That's a key finding.
Definitely. And how did that compare to targeting the age hypomethylated sites in T cells?
Much less happened there. No clear, stable changes at the targets themselves and only moderate bystander effects.
So it really seems like targeting those sites that gain methylation with age has a more potent and stable effect on the wider network, at least in T cells.
Seems that way. And they did account for changes just from culturing the T cells, right, with controls?
Yes, they had controls cultured for the same time. They also noted an enrichment of polycomb targets these H3K27E3 regions among the bystander sites in T cells, which might point towards specific regulatory pathways being involved.
Okay. Now, the big question. What did all this do to the epigenetic clocks in those T cells?
Right. So when they targeted the age hypermethylated sites, they saw an acceleration of epigenetic age according to several clocks.
Acceleration, how much?
Quite a bit in some cases, up to 10 years older prediction.
Whoa. Okay, but hang on. We need to be careful here, don't we?
Exactly. This is the critical caveat. Many of these clocks include the very CpG sites they were targeting in their calculation.
Right, so if you manually increase methylation at a site the clock uses to predict older, the clock is obviously going to predict older.
Precisely. It doesn't necessarily mean the cells are biologically aging faster in a functional sense. It might just be an artifact of directly manipulating the clock's inputs.
It's like changing the number on the warning light on the dashboard doesn't mean you fixed or broke the engine itself.
Perfect analogy. So the clock results are interesting, but we absolutely cannot jump to conclusions about causality here. The effects on clocks were also less clear when they targeted the hypomethylated sites, matching the weaker methylation effects.
And did the bystander effects push the clocks in one direction?
Well, they saw both hyper and hypomethylation happening at bystander sites simultaneously, suggesting the overall bystander effect wasn't necessarily directional for age prediction. But they did find a strong correlation. The more strongly associated a CpG site was with T-cell aging normally, the more likely it was to be affected by the bystander changes.
Reinforcing that connection to the actual aging process, even if the clock readout itself is complicated.
Exactly. Okay, lastly, the MSCs. What happened there?
Mesenchymal stromal cells. They saw modest methylation increases at some target sites after 11 days of targeting the hypermethylated region.
And the bystander effects in MSCs.
They saw increased methylation at bystander sites known to change with long-term culture, which is often linked to senescence or cellular aging.
Okay, so connecting to culture-induced aging markers.
And similar to T cells, both the increases and decreases in bystander methylation were enriched at CpGs known to change with age in blood, and also those strongly correlated with age in T cells. Again, particularly those sites that gain methylation with age.
So the general pattern holds across different primary cell types. Targeted editing influences other age-related sites, especially those gaining methylation.
Seems to be the case. It points towards a broadly conserved network interaction.
So this brings us back to that really critical point about the clocks. Let's just really nail this down. Why are these clocks, especially the fancy neural network ones, potentially problematic for proving causality?
Right. It's super important. These clocks, especially machine learning or NN-based ones, are essentially pattern recognition machines. They're trained on huge data sets. Here's the methylation pattern. Here's the person's age. They learn which CpG sites are the best predictors of age.
They find the correlations.
Exactly. They find the features, the CpG methylation levels that correlate most strongly with chronological age. Now, if your experiment directly changes one of those key predictive features...
The prediction will change, by definition.
Precisely. The clock sees the methylation level go up at site A, which its algorithm learned is associated with being older, so it outputs an older age. But that change tells you nothing definitive about whether the underlying biology of aging has actually been altered.
It's just reflecting the change you forced upon one of its input variables.
Yep. The speedometer analogy holds. You can move the needle, but it doesn't mean the car's speed changed. These clocks are currently validated as predictors, maybe risk indicators, but not as biomarkers of intervention efficacy for changing the aging rate itself.
We can't use them, at least not naively, to say we made the cells older or we made them younger just because the clock number changed after we directly edited the sites the clock looks at.
Exactly. Extreme caution is needed when interpreting clock changes in these kinds of editing studies.
Okay, so wrapping this up then. The main takeaway from this paper seems to be that, yes, targeted epigenetic editing at one age-related spot can trigger these widespread, reproducible bystander changes across the genome.
And these changes aren't random. They preferentially hit other age-related sites, suggesting this interconnected network is real, possibly mediated by 3D genome structure.
And this network effect can alter epigenetic clock predictions.
But this doesn't automatically mean we've found a way to control biological aging or that the clocks are measuring a true change in aging rate in this context.
Right. The functional consequences, whether these changes are good, bad, or indifferent for health and lifespan, are still totally unknown. This paper shows the connections, but not the path to a therapy yet.
Absolutely not. We need in vivo studies in animals to see what actually happens functionally. We need multi-omics to see how these methylation changes affect gene expression, proteins, everything else.
And we still need to figure out the exact mechanisms. How does the signal spread? What role do chromatin, histones, other factors play?
Loads more research needed. This opens doors but doesn't walk through them.
So maybe the final thought for our listeners is this. Given this incredible interconnectedness, this network effect revealed by the study, how do we even begin to think about intervening safely and effectively? If tweaking one spot has effects all over, how do you target the process of aging without just, messing with the markers or causing unintended consequences elsewhere?
That's the multi-million-dollar question, isn't it? It highlights the complexity we're up against. Really fascinating work, though.
Definitely makes you think.
Yeah.
Well, thanks for joining us for this deep dive.